Welcome to the deep dive. Today we are wading into a really fascinating ecological mystery.
We are indeed.
We're looking at one of the world's most, well, frankly, destructive biological invaders,
Yeah.
the American bullfrog, Lithobates cansmianus.
A notorious one. Everywhere this massive frog establishes itself, you tend to see native amphibian populations just plummet.
But here's the puzzle, right? Not all native species crash. Some seem highly susceptible, they just disappear. Others, though, they seem to kind of shrug off the presence of this, well, ecological bully.
Exactly, and that's the core question.
So our deep dive today is tackling that exact question. We're analyzing some crucial research led by Everts titled Phenological Mismatches Mitigate the Ecological Impact of a Biological Invader on Amphibian Communities.
Yeah, that title really sums it up. We know invasive alien species, IAS, are a huge problem, ecologically, economic.
Massive costs.
The damage they cause globally is often compared to, major natural hazards. But with the bullfrog, we've been missing this critical piece. Why, at the community level, does it seem to impact some species so much more than others? What's the mechanism?
Okay, let's maybe set the scene first. Just how dangerous is the bullfrog? It's not just a bit of a pest, is it?
Oh, definitely not. It's a full spectrum threat. First, it's a major predator, gape limited, which basically means
It eats anything that fits in its massive mouth.
pretty much tadpoles, other frogs, fish, even small snakes or birds sometimes. Then there's competition. It competes fiercely for food, for shelter.
And maybe worst of all, disease.
Yes, it acts as a carrier, often without showing symptoms itself, for really devastating amphibian diseases like chytrid fungus brood and ranaviruses. These can wipe out native populations.
So it's a predator, a competitor, and a disease vector, a triple threat.
Exactly. But despite knowing all that, we've historically struggled to pinpoint the specific, interspecific mechanisms, the repeatable reasons why one species vanishes while another living right alongside it persists.
And without nailing down that mechanism, our ability to predict future impacts is limited.
Precisely. Our efforts to do preventative horizon scans, basically risk assessments for potential future invaders, they're inherently a bit flawed if we don't understand why certain species are vulnerable.
Okay, and that's where this paper comes in. The researchers, they sort of jumped in with a central hypothesis.
A really interesting one.
They asked if the key protective factor isn't necessarily about, say, genetics or specific habitat needs, but simply about timing.
Yeah, could a temporal mismatch, basically, a separation in their key life events, specifically breeding cycles, could that actually shield native species from the bullfrog's worst impacts?
It sounds elegant, simple, almost, but testing, it must have been tricky.
Oh, absolutely. It required solving a pretty big data problem. I mean, traditional ways of monitoring wildlife, like visual surveys or trapping, they're useful, but
They have limitations.
big time.
Yeah.
Especially when you're trying to quantify something like bullfrog abundance across a large area. They can be secretive, hard to spot consistently. You get observation errors, patchy coverage. It's tough.
So how did they tackle that? This study had a novel approach, right?
It did. They used a large-scale space-for-time approach. They looked at 107 different ponds across northern Belgium.
Okay.
And these ponds represented the entire bullfrog invasion gradient. So some ponds had no bullfrogs, some had a few, some were heavily invaded.
So by looking across space, they could kind of infer the impact over time without needing decades of data from one place.
Exactly. It gives you snapshots of different invasion stages simultaneously.
Yeah.
And to get really good standardized data from all 107 ponds, they turned to environmental DNA.
eDNA.
Yeah.
eDNA. This is where it gets really interesting, I think. It lets you detect species just from traces of their DNA in the water.
Yep. And they used it in two clever ways here.
Okay, maybe break that down for us. How did they use these two different DNA tools to measure both the invader and the natives?
Sure. So think of it like a two-pronged DNA attack. First prong, target the invader, the bullfrog. They used a technique called quantitative barcoding, specifically something called DDPCR.
Quantitative.
Yeah,
So measuring how much?
exactly the point. This tool let them accurately measure the absolute concentration of bullfrog DNA in each water sample. And that concentration is a really reliable proxy for the actual abundance or density of bullfrogs living in that pond.
Okay, so they could get a real number on the bullfrog pressure in each pond, not just present or absent.
Precisely. A solid number for the threat level. Now, the second prong focused on the native community. For this, they used metabarcoding.
Metabarcoding. That's one that looks for everything at once, right?
Sort of, yeah. It targets a broader genetic marker in this case, the 12S rRNA gene that's common to lots of vertebrates, including amphibians and fish.
So what did that tell them?
It didn't give them an accurate count of every single native newt or frog. That's still technically challenging with metabarcoding. But what it did give them was a comprehensive, standardized list of which native species were present or absent in each pond.
A reliable catalog of the native community.
Exactly. So quantitative data on the invader, presence-absence data on the natives.
And then they needed the timing aspect, the phenological.
Right. That was the final piece of the puzzle. They integrated decades of citizen science data, observations from platforms like Warnemingen.b going back to 1996.
Wow, 26 years of data.
Yep. And that huge data set let them pinpoint the average timing, the mean Julian date for key reproductive events, like when eggs or larvae were typically seen, for both the native species and the bullfrogs.
So marrying that precise ecological timing data with the quantitative invader data, that was the key innovation here.
Absolutely. That's the genius move, and the results from their Belgian field study. Pretty stark.
What did they find?
A really significant negative relationship. The higher the bullfrog eDNA concentration, basically, the more bullfrogs, the lower the overall native amphibian species richness. Fewer native species survived where bullfrogs were dense.
A clear impact then. But did it show that species-specific vulnerability you mentioned earlier? Did some species suffer more than others?
It did, very clearly. They found the community structure was nested.
Nested. What does that mean exactly?
It means the loss of species isn't random. Imagine the community in an uninvaded pond is like a full set of Russian dolls. As bullfrogs invade more heavily, you don't just lose random dolls, you lose the smallest one first, then the next smallest. The communities in heavily invaded sites are just predictable subsets of the richer communities.
Ah, I see. So the bullfrog invasion follows a pattern, systematically knocking out certain species first.
Exactly. It strongly indicates species-specific exclusion.
And could they tell which species were getting knocked out first? The smallest dolls.
They could. The analysis showed bullfrog abundance significantly hammered 3 specific newt species: the smooth newt, the alpine newt, and the palmate newt. They seem to be the most vulnerable.
Okay, so those newts were clearly struggling. But what about the species that weren't affected?
That was just as revealing. 2 very common native amphibians, the common toad, Bufo bufo, and the common frog, Rana temporaria, were found to be completely unaffected. Their presence didn't decline at all, even in ponds packed with bullfrogs.
So stark differences in vulnerability.
Definitely. And to make sure this wasn't just some local Belgian phenomenon,
They went global, right? They did a meta-analysis.
they did a huge one. They synthesized data from 56 different cases, drawn from 20 unique studies all around the world where bullfrogs have invaded.
And did the global picture match the Belgian findings?
Perfectly. The meta-analysis confirmed a strong negative relationship between the degree of phenological mismatch, the difference in breeding times, and the bullfrog's impact globally.
And they actually put a number on it, didn't they? A threshold.
Yes, and this is so valuable for conservation. The key finding was this. Native species whose breeding phenology differed by 6 weeks or less from the invasive bullfrog's breeding time were statistically much more likely to be absent from invaded sites.
6 weeks or less overlap, and you're in trouble.
But if your breeding time was more than 6 weeks different from the bullfrogs,
You were likely okay. Wow. 6 weeks. That's a really concrete margin.
it really is. It provides a clear threshold for vulnerability based on timing.
So let's unpack why. Why does this 6-week window matter so much? What's the mechanism?
It comes down to the bullfrog's own biology and timing. Being warm adapted, it's a late spring and summer breeder.
Right, needs warmer water.
Exactly. In their Belgian study area, peak reproductive activity was around April-May, stretching into summer.
And those unaffected native species, the common toad and common frog.
They're the opposite. They are classic winter breeders. Their peak activity is way earlier, February and March, often when there's still ice around.
Ah, so they're basically done with the most vulnerable parts of their life cycle before the bullfrogs really get going.
Precisely. Think about the pond as a shared resource, like a road. If one species commutes, breeds, and has vulnerable larvae at 6 a.m., and the bullfrog commutes is actively hunting and breeding at 6 p.m.
They use the same road, but they never actually meet.
Exactly. By the time the large, hungry adult bullfrogs emerge from hibernation and become really active in late spring, the main reproductive period for those winter breeders is finished, their eggs have hatched, and often their tadpoles have already metamorphosed or are close to it.
So the most vulnerable stage, The eggs and small larvae that bullfrogs would easily predate or compete with or transmit diseases, they're just not physically present in the water at the same time as the active adult bullfrogs.
Correct. They use the pond sequentially. This temporal separation limits co-occurrence, and that's the key.
Because no co-occurrence means no interaction.
Right, it mitigates all three main threats at once. Less chance for direct predation by the big bullfrogs, less competition for food and space when the native larvae are tiny, and less opportunity for diseases like ranavirus to jump between them during peak vulnerability.
The timing mismatch acts like a shield.
A very effective one, it seems. Phenological overlap is essentially required for these negative interactions to occur at the most damaging level.
Okay, so what are the big takeaways here? What does this mean for conservation, for how we manage invasive species?
Well, the major practical implication is that this timing factor, this phenological overlap, it's clearly a really strong predictor of invasion impact, certainly in temperate systems like the ones studied, and it's something we've often overlooked.
We focus so much on where invaders are, maybe not enough on when they're active relative to native species.
I think that's fair to say. So the researchers strongly recommend that temporal alignment, the degree to which an invader's key life stages line up with a native species' vulnerable periods, should be formally integrated into how we assess invasion risk globally.
So alongside things like how many invaders there are, or how widespread they are.
We also need to ask, do their active periods clash with the vulnerable periods of native species? Knowing when seems just as crucial as knowing where or how many.
That feels like a really valuable addition to the toolkit. Now, you mentioned earlier a limitation regarding the native species data. The metabar coding gave presence-absence, not density.
Right?
Why does that mean the negative impacts reported in the study are likely conservative?
That's a really important point about interpreting the results, because they measured native impact mainly as complete local absence, the species being totally gone from the pond.
They might miss less severe impacts.
Exactly. Population decline almost always happens before a species is completely wiped out locally.
Hmm.
A species population could crash by, say, 90%, making it ecologically irrelevant, maybe functionally extinct in that pond. But if even a few individuals hang on.
The metabar coding would still register it as present.
Correct. So the study could only definitively flag the most extreme outcome, total exclusion. The actual ecological damage occurring before that point, the declines in abundance below that 6-week threshold, is likely much greater than just the reported species absences.
Got it. So the 6-week threshold is likely where things get really bad, leading to local extinction, but the damage probably starts even with slightly more mismatch.
That would be the logical assumption, yes. The quantitative approach for the bullfrog itself using ddPCR was a real strength, though, giving that precise pressure metric.
Okay. So wrapping this up, this deep dive really highlights that the breeding schedule, the simple when of a species' life, is a critical, quantifiable factor in predicting its vulnerability to a major invader like the bullfrog.
It really does. And if we take a step back and think about the broader context of global change, well, the implications get even bigger, maybe even a bit worrying.
How's
Climate change. We know it's rapidly shifting phenology, the timing of life cycles for countless species worldwide. Plants are flowering earlier, insects emerging sooner, birds migrating at different times.
And amphibians are breeding earlier in many cases too, right?
They are. But here's the crunch. What if native species and invasive species respond differently to that warming? What if the early breeding native frogs shift their timing significantly, but the warm adapted bullfrog responds less or differently?
You could lose that protective mismatch.
Exactly. That currently protective 6-week buffer, it could shrink or disappear entirely. You could end up unintentionally synchronizing currently safe native species with the peak activity of invaders like the bullfrog.
Turning that temporal shield into a sudden, catastrophic vulnerability.
It's a real possibility. The dynamics of phenological synchrony, or lack thereof, could fundamentally change conservation outcomes in the coming decades. What protects a species now might not protect it in a warmer future.
Which leaves us, and you the listener, with a really important question to ponder. We see how this natural time difference currently shields some species. But given that climate change is scrambling these biological calendars, how might future warming unintentionally erase that protection? Could species we currently consider safe because they breed early suddenly find themselves overlapping with invaders like the bullfrog? And crucially, what proactive conservation steps could we even begin to think about now to prepare for the potential loss of this natural time-based defense? Something to think about for sure.